Hello, everyone, and welcome to today's webinar in the CERDP and ESTCP webinar series. My name is Rula Deeb. I'm a senior principal at Geosyntac Consultants in California. I'm the coordinator of the webinar series on behalf of CERDP and ESTCP. It is my pleasure to facilitate today's call. The webinar will consist of a brief overview of CERDP and ESTCP, followed by technical content, which details results from DoD-funded research on the roles of soil microbial communities in ecosystem restoration. First, Dr. James Bever from the University of Kansas will talk about the restoration of native grassland ecosystems by reintroducing native soil fungi, which improves the establishment of desirable native plant species and suppresses invasive plants. Second, Dr. Nichole Barger from the University of Colorado will discuss restoring arid and semi-arid lands through seeding soils with enhanced biocrust. Q&A sessions for each speaker will be held at the end of their respective presentations. On this slide, we have provided a few suggestions in case you experience technical difficulties with the broadcast platform. Firefox, IE, or Edge are the most compatible browsers to use. If you lose the content on your screen or if your screen freezes, try keying Control F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to have technical difficulties, please call into the conference line shown here. You may also submit a comment using the chat box. Please use that chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions that will be posed to the speakers. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A periods to submit your questions. In fact, we do encourage you to submit questions in advance of those sessions. When submitting your questions, please make sure to add your organization name at the end of your questions so that we can identify you during the Q&A session. With that, I would like to introduce Dr. Kurt Preston, who is the CERDP and ESTCP Program Manager for Resource Conservation and Resiliency. Dr. Preston has tracked his career between civilian university and military positions. Prior to joining CERDP, he was a faculty member and research administrator at the University of Nebraska-Lincoln, where he led faculty development efforts to improve research competitiveness. In this position, Kurt also worked with technology transfer personnel, academic departments, and colleges to build university research capability. In addition, Dr. Preston served as a member of the Chief of the Army Corps of Engineers Environmental Advisory Board. Kurt?
Hey, thank you, Rula. I am happy to welcome everyone to today's CERDP and ESTCP webinar. So CERDP is the Strategic Environmental Research and Development Program established in 1991 by Congress as a partnership between the Department of Defense, the Department of Energy, and the Environmental Protection Agency. CERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. CERDP funds both fundamental research as well as advanced technology development that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate the innovative environmental and energy technologies These investments capitalize on past investments under CERTIP or other research programs and are designed to transition technologies out of the laboratory and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. CERTIP and ESTCP are complementary programs, with much of CERTIP research occurring at the lab and pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting laboratory efforts are conducted. There are 4 program areas in CERTIP and 5 in ESTCP. The Installation, Energy, and Water program area is only found in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Resiliency, and Weapon Systems and Platforms, are found both in CERTIP and ESTC, and their programs managed jointly by a designated program manager. Our webinar today is focused on research and demonstrations that were conducted under the Resource Conservation and Resiliency program area, which has essentially 3 main areas of research focus. 1, natural resources, 2, resilience, and 3, air quality or wildland fire. Our webinar series highlights research and demonstration efforts from each of the 5 program areas. As you can see, upcoming webinars will cover a broad range of topics, including the treatment of emerging contaminants in the subsurface, the development of new non-toxic and sustainable paints to prevent corrosion, tools for vapor intrusion and mitigation, and understanding underwater munitions mobility and behavior on the beach face and in shallow, muddy environments. The next resource conservation webinar is on June 20, and it will focus on adaptation strategies to address uncertainties around climate change. You can find more information about upcoming webinars at this link, and registration is live for webinars through the end of the calendar year. I would like to remind you that a copy of the presentation of today's session can be downloaded from our webinar webpage. And we would appreciate it if you could please take a moment to complete the survey that will pop up on your screen at the end of this webcast. Finally, I am pleased to announce that the CERTIP ESTC Symposium will be held again this coming December in Washington, D.C. The 3-day event will showcase the latest technologies that enhance DoD's mission through improved environmental and energy performance. Registration information is available on the CERTIP and ESTCP website. Finally, I sure hope you enjoyed today's webinar.
Thank you so much, Kurt. It is now my pleasure to introduce our first speaker. Dr. James Bever is a professor in the Department of Ecology and Evolutionary Biology at the University of Kansas. Jim's research focuses on understanding the influence of microbiomes on plant productivity and diversity. He moved to the University of Kansas in 2016 when he was appointed as a distinguished professor. And prior to this, he was a professor at Indiana University, where he also co-chaired the Evolution, Ecology, and Behavior faculty. Jim has co-authored 150 scientific articles, and he's received multiple fellowships, including the Distinguished Fulbright Fellowship. He received his bachelor's and master's degrees in biology from the University of Illinois and the University of Michigan, respectively, and he received his doctoral degree in botany from Duke University. Jim, please proceed.
Okay, thank you, Rula. So I want to acknowledge my collaborators on this project, Gail Wilson, Karen Hickman, and Peggy Schultz, and the team of researchers from University of Kansas, Oklahoma State, and Indiana University that made this work possible. And thanks, SERDP, for the support for this research. So in this talk, I'm going to give an overview of arbuscular mycorrhizal fungi, describe evidence that the community composition of these fungi are degraded by soil disturbance, and then present evidence that native and desirable plants really need these fungi, and that reintroduction of native mycorrhizal fungi can improve restoration. So we'll start with an overview of the project's motivation. So native grasslands are highly diverse, and they include many plant species that have desirable characteristics and are good hosts for other herbivores, butterflies, pollinators that are valued. And with disturbance, with military training or tillage or any other disturbance of the soil, these communities often are replaced by non-native invasive species, so species with less desirable characteristics. And also, the soil community may be disturbed during that time. And so what we're going to investigate is whether the soil community, and particularly arbuscular mycorrhizal fungi, which these are spores of eight species of mycorrhizal fungi presented here, whether they're disturbed and whether reintroduction of the native composition of these fungi can improve restoration of establishment of the native species. So arbuscular mycorrhizal fungi form associations with nearly all plants. And so roots go into the soil and they're infected by these hyphae of these fungi, and they form these arbuscules in the cortical cells of plant roots. And then they have hyphae which radiate through the soil and bring in very difficult to access resources such as phosphorus. And they can have a variety of other effects on plants, including altering their defense. And through that, they promote plant growth. They also stabilize the soil. And so these are beneficial organisms that have high value for ecosystem function. So to evaluate whether the community of mycorrhizal fungi is degraded with disturbance, we worked at 3 military installations, the retired Air Force Base in Illinois, Chanute Air Force Base, Fort Riley Army Base in Kansas, and the Tinker Air Force Base in Oklahoma. And we supplemented sampling of these sites with 16 other sites across Kansas, Oklahoma, Missouri, and Illinois. And at each of these locations, we sampled native uninvaded grasslands and nearby degraded grasslands. And we sampled them over 2 years. At each of these sites, we evaluated the plant community composition, and we took soil samples, and from these soil samples, we characterized the community of mycorrhizal fungi by sequencing the ribosomal RNA gene. So this is a figure presenting the results that we found. And so on this axis, we're arranging different species of mycorrhizal fungi that we saw in across these communities. And these bars, the bars that are to the right are species that were differentially abundant in the remnant undisturbed sites, and the bars on the left are differentially abundant on the disturbed sites. The solid bars are significantly different from random. And so what you can see is that there are many species of mycorrhizal fungi that are represented here on the right that are abundant in remnants and then are lost with disturbance. And this is generalizing across all of the sites that we sampled. And there are other species which are differentially abundant and accumulate with disturbance. And so this is evidence that the community of mycorrhizal fungi is degraded. We're losing these fungi, these native species of fungi, and we're accumulating these weedy fungi with disturbance. So we also sampled across a rainfall gradient, and so we can look for differentiation along that rainfall gradient. And because of the strong differentiation, we looked within the remnant undisturbed grasslands and the disturbed grasslands separately. And so that's what we're looking at here. So in the native grasslands, we see strong differentiation. So we have many species which are differentially abundant in the east, other species which are differentially abundant in the west. And this is what we would expect if the differentiation of the mycorrhizal community aided plant adaptation to differences in climate, differences in precipitation along that east-west gradient. In the disturbed sites, in the post-disturbance sites, we don't see that differentiation. We see an abundance of species in eastern disturbed sites, and it's less clear that there's a differentiation in the west. And so we're losing the differentiation of the mycorrhizal community with disturbance of these grasslands. So the implications of these results is that, first, military training can degrade the community of mycorrhizal fungi. Native mycorrhizal fungi can be important for adaptation of plants to climate, and degraded mycorrhizal fungi are not likely to facilitate adaptation to climate. So we then want to know if the extent to which mycorrhizal fungi mediate the success of native plants, and does the degradation inhibit our ability to reestablish them? And so we want to know if there's differential response of non-native species such as tall fescue between native species. And we break the native species into two groups: early successional species which establish easily on disturbed soil, and big blue stem or late successional native species such as big blue stem, which really have difficulty establishing soon after soil disturbance. And so our approach is to look at suites of species of these three categories. And I'm going to show you two experiments. In the first experiment, we looked at 12 species, 6 early successional native species and 6 late successional native species. And we evaluated their response to several species of mycorrhizal fungi. And so this figure is a little bit complicated initially, so I'm going to talk you through it. So on this axis, we're looking at the percent change with inoculation of mycorrhizal fungi of each of these plants. So, for example, and then these are groups are the different plant species. So there's 6 different groups for early successional, 6 different groups for late successional species. And then the different colors represent the different response from the different fungal species. So this species here, which is Allium sinuum, really grew well with this species, which is Chlordioglomus luteum, but it didn't grow at all well with this species, another species of mycorrhizal fungi. So what we see is that the native late successional species are very responsive to mycorrhizal fungi, but also very sensitive to mycorrhizal fungal species composition. They grow well with some, but not with others. While the early successional species don't benefit in this experiment, and they don't differentiate, they don't have differential responses to the different species of mycorrhizal fungi. So this is consistent with the composition of mycorrhizal fungal species mattering more to late successional species than early successional species. And to compare this response to non-native invasive species, we repeated the study, including several non-native species, as well as representative early and late successional species. And what we find is, again, late successional species are responsive to mycorrhizal fungi, and it matters, the composition of the fungus matters a lot, while early successional species and non-native species don't respond differentially to mycorrhizal fungi. And this is consistent with the degradation of the mycorrhizal fungal community, contributing to dominance of non-native species and limiting the success of late successional native species. So to test whether we can reintroduce the native mycorrhizal fungal community as a way of aiding establishment of the desirable late successional native plant species, we conducted 5 different experiments distributed across 3 military bases: the Chanute Air Force Base, the Fort Riley Air Force Base, and the Tinker Air Force. The Fort Riley military base, Army base, and the Tinker Air Force Base. And so these experiments were conducted on sites that were dominated by non-native, undesirable species. And at each of these sites, we cleared the vegetation by solarization or tilling or herbicide or a combination of that. And we used 3 different inoculation treatments. We introduced whole soil from nearby native sites, nearby native prairies, or we isolated mycorrhizal fungi from those nearby prairies, or we had uninoculated controls. And these inoculation treatments were conducted by inoculating sterilized soil and then planting into that representative native plants, late successional native plants, into this soil, establishing the plants and then putting them out into these plots. So across these 5 experiments, we planted out 1,800 different plants of 9 different native plant species. And then all of these plots were seeded with a native plant mix. We then monitored the establishment of these plots over the next several years. So what we found is that here we're looking at the first year survival. Survival was high in plots that were inoculated with mycorrhizal fungi or whole soil, whole native soil. And they were higher in these treatments than in the control. There wasn't a difference between the mycorrhizal and the whole soil, which suggests that the effect is mainly due to the mycorrhizal fungi. We see a similar difference in survival to the second year, and for the plants that survived, we have greater growth of the plants when they're inoculated with native mycorrhizal fungi. So this is consistent with inoculation facilitating the establishment of native species and their growth. So to give you a representation of what this might look like, if you started a restoration with 10 plants of grasses and composites and legumes, then monitored their persistence over the next several years. By the second year, you'd have much more a greater abundance of the natives established in the inoculated plants, and this gets exacerbated in the third year. And of the plants that survived, they grew bigger with the native inocula. And so together, this brings the community closer to the target of the restoration, closer to the target, the representation, the composition of the native prairie. So does inoculation suppress non-native species? To look at this, we conducted a second experiment which distributed the inoculum over the plot more generally. So in this, we again have 16 different plant species within the plot of 4 different mycorrhizal species, as well as a mixture of all of these inocula and a sterile control. And then the plots were again seeded with a diverse mix and monitored over the next several years. And so this is the survival results. We again see that plants inoculated with the native mycorrhizal fungi grow better. in here we see that the diverse mixture of native fungi is as well or better, as well or better than the individual species of mycorrhizal fungi. So that's true for plant survival and the growth of the plant during the first year. And when we look at the composition of these plots, we see that the plots inoculated with a diverse collection of native mycorrhizal fungi have higher representation of desirable plant species and therefore lower representation of undesirable species, while the uninoculated controls has a high proportion, 90% of the cover is undesirable species, undesirable plant species. So we then are able to demonstrate that mycorrhizal fungi is degraded by disturbance of the soil. This degradation promotes plant invasion and inhibits native plant reestablishment. We're able to then show that if we reintroduce the native mycorrhizal fungi, we can increase native plant establishment and inhibit non-native weeds. So the potential benefits of this for management of DoD properties is that better native plant it gives a mechanism for better native plant reestablishment after training activities, decreased non-native invasive problematic weeds could reduce management workload, and we find that evidence that mycorrhizal fungi can improve plant adaptation to climate change, and the establishment of these natives can support other management goals, such as increasing habitat for desirable butterflies. And with that, I thank you for your attention and give the controls over to the questions.
Great. Thank you so much, Jim, for a great presentation. As a reminder, I'd like to remind you guys to submit your questions by using the Q&A box on your screen. We have received a number of questions on this presentation that Jim will answer. So Jim, your work focuses on tallgrass prairie. How well do you think your results will translate to the restoration of other native plants, for example, in the California grasslands?
Yeah, that's a good question. We have done some work in the California grasslands. We find that the native plants are much more responsive to the mycorrhizal fungi, the native mycorrhizal fungi, than the non-native species. And so we think that the same approach can be valuable in that system as well. And we've done a few inoculation studies which support that, but there hasn't been as much work on this done in that system.
Great, thank you. Are native fungi, the ones you described in your work, are they available for purchase?
There's many commercial products advertised for mycorrhizal fungi. It's become fairly popular in some settings, and in several of them, from what we've found, are not very helpful for the native plants. There is some of the isolates that we've developed are available through a company called Microbial. So that's one place if you want to establish the native tallgrass prairie species, the inocula from Microbial would be what you'd want to use.
Great, thank you. Have you ever tried non-native AMF species to improve performance?
We have done a couple of trials with some of these commercial products for the native plants, and in several of them, they inhibited the late sessional native plant growth more so than the uninoculated control. So I can't really recommend many of the non-native commercial products.
Great, thank you. Here is a question from PNNL. What were the experiments that demonstrated resiliency to climate change? Can you elaborate on those?
Well, so we're focusing on the role of the plant microbiome in their establishment, and we had several complementary experiments that looked at the extent to which the plant microbiome mediated their tolerance to drought, and they were encouraging that the differentiation that we saw with the along the rainfall gradient, that the mycorrhizal fungi from the drier areas did support growth in that environment.
Great, thank you. You briefly spoke on your experimental sites occurring on a precipitation gradient. How do you think a heat gradient may affect the fungal community and therefore plant productivity and ecosystem functionality?
It's a good question. We did have some variation in the temperature gradient in the Oklahoma to Illinois transect. It worked out that the temperature wasn't so important for the mycorrhizal fungal composition, but when we started looking at other fungi, they were much more sensitive to the temperature. Sensitive to temperature as well as the precipitation gradient. So that includes saprophytic and pathogenic fungi. So I think that the temperature gradient is going to be very important in determining the importance of mycorrhizal and non-mycorrhizal fungi in plant communities.
Okay, great, thank you. A question from Tetra Tech. Have similar results to inoculation with native soils and AMF complexes been observed in the desert and forested communities in the Intermountain West?
I know of several studies of that, and I think from what I remember there, it's had mixed results, and so I think that there hasn't always been a positive response to the native inoculant in those studies, and I don't know exactly whether it related to the plant that was being compared or the source of inocula on why that was. But I think that in principle, a lot of the very long-lived late sessional plants are responsive, and so it should help in the but that more work needs to be done to demonstrate that.
Thank you. A question from EPA. Are there indirect effects through the influence of AMF on other members of the soil microbial community or soil macrofauna that may impact the plant community?
It's very true. The soil community is very complicated, and the fungi can directly inhibit other fungi. And so, for example, in one experiment where we did use a non-native inocula, there we had evidence that it inhibited the resident fungi from infecting the roots, and that might have been why it ended up not helping the plants. So there's so many possible interactions below ground that we can't monitor them all, and so it's the kind of question that could be a lifetime of research to answer.
Well, those are the best types of research projects. All right. A question there's a lot of questions, Jim. We'll try to get through as much as we can right now, and we'll save some towards the end of the webinar. But this is a question from Ecology and Environment. Are regional or continental sources of AMF important to the success of target site inoculation? I think you alluded to this a little bit, Jim, but if you can elaborate, that would be awesome.
So if I understand the question, it's does this relate to the importance of the inocula being from similar climate and soil types as the target community? Is that, am I interpreting that question correctly? In that case, I think that it is, and we don't know the scale of how local is desirable, and so it would take more work to demonstrate how far from a target site can you get fungi to improve the growth at that target site, but it certainly is going to be critical.
Great, thank you. All right, and these are two questions from industry that are related and have been asked more than once. What is the average deepest depth of plant roots? And can you please explain again how you actually did the inoculation with the AMF?
Okay, so the prairie plant roots can go to meters deep. They can be very, very deep, and so there's no way we can put the fungi in that entire depth. And so what we're relying on is the fungi growing with the plant root as it grows deep into the soil. And the way we conducted this inoculation is what we call nurse plants, where we're basically babying the native fungi by giving them a host plant to grow on, and we start them in the greenhouse for a month. And then once they're growing, we then put them out into the field, and we make sure that nurse plant survives that first month after transplant. We water it that first couple weeks to get it established, and then we let it go. And so the idea is to introduce the fungi into the site by getting that plant established and having the fungus grow with that plant's root system. So
great, thank you. All right, another question from Pacific Northwest National Lab. Do the positive results with the mixed AMF inoculum suggest synergies between the AMF species or just enhance benefits to the native plant?
So what we found in that study and in a couple other studies is that when you put mixtures of mycorrhizal fungal species together, the plant tends to grow as well as the best fungus. And so it doesn't appear that there's synergistic benefits of having many fungi together, just that as long as the best fungus for that plant or for that environment is available, the plant is able to access it. And even though there might be other fungi in the root system, it's able to grow as well as if it was only that best fungus.
Great. And then one last question for you before we transition to Nichole's presentation. And this is a question from the Washington Military Department. Did you notice a spatial distribution in native plant composition corresponding to where you placed your host plant?
Yes, and that's an excellent question. And that was, in this particular experiment, we set out to look at spread of these fungi from those nurse plants. And so we arranged them in a single row. And then we did measure composition at different distances from those rows. And we've sampled the soil at different distances from those rows. And so in these particular studies, we did see some differential effect on plant community composition as we moved away from those rows. And we were able to detect, in the soil, we were able to detect the inoculated fungi at different distances. That story ends up being so complicated to tell because the rate of spread of the fungi varied between the fungal taxa and between the sites. In other studies we've done subsequent to these ones, we did find stronger gradients in plant composition from the points of inoculation, but So I think there's going to be some context dependence on the rate of spread of these beneficial fungi, and we're still in the process of understanding that.
Wonderful. Thank you, Jim, for entertaining these questions. At this point, we're going to move over to our second presentation today, which will be delivered by Dr. Nichole Barger, who is an associate professor in the Department of Ecology and Evolutionary Biology at the University of Colorado in Boulder. Nichole's research focuses on sustainable land management and the restoration of a broad variety of ecosystems throughout the world. She is actively involved in a wide range of international science policy activities. For example, she is serving a 4-year term as one of 10 scientists on the United Nations Convention to Combat Desertification Science Policy Interface Group, which promotes dialogue between scientists and policymakers on a range of topics. Nichole is the current science director at the Nature Conservancy Canyonlands Research Center in Utah. She earned her bachelor's degree from the Evergreen State College, her master's degree from the University of California at Berkeley, and a doctoral degree from Colorado State University. Nichole, please proceed.
Thank you, Rula. So first, I would like to acknowledge the many, many collaborators, graduate students, undergraduates that we had on this project. Most importantly were our collaborators, John Kipp in Fort Bliss, who was important early on with the logistics for this project, and also Russell Lawrence from Utah Test and Training Range. The work I'll be presenting today was not only a collaboration with the University of Colorado, but the U.S. Geological Survey, Northern Arizona University, and Arizona State University. So our agenda is that I'd first like to talk about our objectives. I'd like to talk about then what biological soil crusts are and why they're important, and our approach to restoring biological soil crust communities in Western U.S. arid land soils. And our approach was establishing nurseries and then looking at some of the successes and challenges we had with restoration. Then I'll have some conclusions from our work and be able to discuss some of the benefits to DoD. So in this project, our objectives were to facilitate the recovery of degraded arid and semi-arid Department of Defense lands by restoring biocrust communities. So what we did was establish these we called them biocrust nurseries, where we grew them in greenhouses and in the lab as inoculum testing and supply center for restoration. From there, we were identifying the field methods for applying this inoculum. So in some ways, our talks are very complementary today, because we're thinking about how do we inoculate degraded soils with native microbial communities. And then finally, I'll be presenting some results evaluating soil responses, primarily I'll talk about today, to biocrust restoration in our field experiments. So first, what I'd like to talk about are what are biological soil crusts. So those are what they are communities of microorganisms, a range of lichens, fungi, mosses, and cyanobacteria that really colonize the top few millimeters of soil surfaces in Western U.S., not only Western U.S. Dry land landscapes, but across the world. So it's been estimated that approximately 17% of terrestrial land surfaces are colonized by these biological soil crusts. And where you can really see them is in these plant interspace environments. So as we lose plant canopy cover, the interspaces between plant canopies in these more desert environments are colonized by these diverse microbial communities. So biological soil crusts are important because, first, some of the greatest importance is the stabilizing of soil surfaces. So in this bottom right corner, you can really see with this scanning electron microscope photograph the filamentous cyanobacterial and even mycorrhizal or fungal communities that really bind some of these soil particles together and really stabilize those top 2 millimeters of soil. And also they provide soil fertility. So that, at the surface, there are photosynthetic organisms that provide carbon to the system. And also there's a lot of nitrogen fixing communities that fix atmospheric nitrogen and contribute to enhancing the nitrogen in the soil. And as you can also see with some of these photos, you can imagine how the topography of the surface and the water holding capacity could really influence the hydrologic cycles in these systems that are heavily colonized by biological soil crust. So what we know is that not only military activities, but a broad range of soil surface disturbing activities, such as livestock grazing, oil and gas development, recreational activities on these soils can have a pretty dramatic impact on biological soil crust communities, not only in the short term, but over the longer term. So this is just an example here of military training impacts on soil disturbance, results in biocrust community declines, and then subsequent changes in these ecosystem level effects that I mentioned, such as soil stability, soil fertility, and hydrology. And this is a really interesting photo here. This is from these are tank tracks from Operation Desert Strike, and these tracks would date back to approximately 1964. This is in the Mojave Desert, and what you can see here is that there's still evidence in terms of biological soil crust community recovery, but in the year 2000, when this photo was taken, that of still significant impacts to these biocrust communities. So these potential impacts can affect up to 70% of Department of Defense installations in the dryland west regions. So when we look at the actual functional groups and how long it takes for them to recover, here are some data from that same study where we can look at the cyanobacteria. And what we can see after 55 years is that 46 to 65% of those communities have recovered. And so we consider those cyanobacteria more early successional community recovery, whereas the lichen community really takes a much longer time to recover, in the range of 3 to 6%. So what we did in the study, we chose research sites in the Great Basin at Utah Test and Training Range, and also in the Chihuahuan Desert at Fort Bliss. We're also showing just all the desert regions across the West that this is just a prevalent land type, and the opportunity for large-scale restoration is fairly infinite in this area. And so what we did first in the first years of our project was establish these biological soil crust nurseries. We really had to think about how do we grow inoculum to then inoculate soils for restoring these microbial communities. So our first approach, and this was working with the microbiology lab, Ferran Garcia Pinchel's lab at Arizona State University, is that we took some of these native cyanobacterial organisms from those sites that I just showed, from the hot desert site in the Great Basin, or a hot desert site in the Chihuahuan Desert, and our cold desert site in the Great Basin. We first isolated these early successional cyanobacterial organisms because we really wanted to target those, because those are the organisms that are really going to respond first. So these were isolated, and then what we had to do was increase their biomass. So for some of the organisms, that wasn't a problem. For some of the cyanobacteria, we could just scale up and give them time to grow. And what we came down to was an approximate 3 to 4 months that it would take to scale up the biomass we needed for our study. Interestingly, there were some of the more important early successional cyanobacteria really were problem children. That what we saw is that they needed substrate to grow on. So this photo in particular for the lab grown, what I'm showing here are these Microbial species that are growing on these filter paper, and that's the only way they wanted to grow. And then what we needed to do is, we grew out, I don't know how many thousand plates of these that were then dried, put through a paper shredder. This is a pretty innovative idea on the part of a graduate student, shredded into these small pieces, and then mixed with some native soil as the inoculum. So this is our mixed isolate approach to inoculation that was all lab grown, very labor intensive. So our second strategy, as you can see here, is this greenhouse grown. And so what we did here with the greenhouse grown is that we took small samples from our field sites and we inoculated these small containers in the greenhouse and let them grow out for a period of 3 to 4 months, adding water, nutrients, and light to try and increase their biomass in that way. And so these are our two, as I'm going through the data, you can remember there's the lab grown and the greenhouse grown approaches. So while we were working on how to grow all of these biological soil crust organisms, at the same time we were conducting a series of field trials where we were trying to figure out, what is the best way that once we have inoculum developed to add it in the field. And this is, and our question was, how do we modify the habitat that this inoculum will be successful? So what we did is we modified the habitat by adding shade, adding water, adding nutrients, and we also knew that in these highly degraded sites, soil stability was going to be a big issue, because if you have highly unstable soils and we add a BioCrust inoculum on top of that and it just gets buried, if those photosynthetic organisms get buried by highly mobile soils, we'll just experience a lot of mortality. So I put a red box around the soil stabilization because we knew that this was important, and we knew that that it was probably an important strategy going forward, especially in highly degraded mobile soils. So from these field trials, what came out of this is that, out of all of these field trials, I just want to mention this. It's not a strategy that went forward in our larger scale experiments, but it was something really interesting that we found out. So what I'm going to explain this graph, and you'll see this a few times, this micrograms of chlorophyll A per gram of soil is just an index of photosynthetic, you can think about it as photosynthetic activity and the level of cyanobacteria colonization by BioCrust on soils. So what I'm showing here is that this scraped soil just means that when you remove the BioCrust, this is how much chlorophyll A that we have in the soil, just as an endpoint. And then you go far over to the other endpoint of what does an intact biological soil crust look like. And this is how much in general, in terms of micrograms for chlorophyll A we had in the site. So natural recovery means that in year one, we just scraped it, scraped the soil, and then we watched it recover naturally. We didn't do anything. We didn't add inoculum, we didn't shade it, we added no water. And this is what natural recovery looked like in that system over those 3 years. The second strategy that we looked at was shading. And so what we see after 3 years, when you compare to the natural recovery, we get this nice little bump in the chlorophyll A. And what most interestingly, after 3 years, the shading plus inoculum really reflects now that intact biological soil crust. So I wanted to point this out to show that we do have strategies to be able to enhance biological soil crust recovery. However, we chose not to go forward with this shading because we were very, as you can imagine, reluctant to tell land managers and DOD managers to shade all of their soil. We didn't think that this was realistic, but we also kept this in mind in thinking about, how do we think about shade? And really what the shade is doing in here is that it's decreasing the stress of water loss and the stress of UV. So we've been thinking a lot about how do we decrease stress without providing shade to large landscapes, because really where we want to think about is getting to large-scale restoration. So very quickly, before I talk about our field experiments that we ultimately did, our multifactorial field experiments, we went forward with 3 different types of inoculums. So I talked already about our greenhouse grown and our lab grown, but we also wanted to compare it to the field collected inoculum, right? What you would get from the field. So we talked a lot about the trade-offs of the different types of inoculums. So the biggest thing is our over-harvesting risk. So if we were to go forward with trying to restore crust by collecting crust from somewhere else, you can imagine that the risk of over-harvesting and just disturbing another biocrust community would be very high. Whereas the greenhouse grown is medium because we are still getting samples from the field, but the risk of over-harvesting in the lab is very low because we have these cultures that are in the lab that we can then scale up the biomass. However, the trade-offs is that when how these different types of inoculum are adapted to the local environment. So the field collected is right from that site. It can be, it's highly adapted to that environment. The greenhouse grown has probably a medium level of adaptation, is that it's brought into the greenhouse, these organisms, for a couple months. They've experienced lessening of resource limitation, but there's the risk there of just having a little bit of not being adapted to being in that harsh environment anymore once they've been in that really nice greenhouse environment. Whereas the adaptation of the lab grown, it would be very low to that local environment. So the risk here is that you take lab grown and greenhouse grown and put it in the field, and it just doesn't do that well because it's not very adapted to those harsh conditions. And then the time and resource investment. So the field collected is very low. You can just go collect from another site and inoculate to a different disturbed site. Greenhouse grown takes far more time to grow these biocrusts in the greenhouse. And the lab grown, at this point in our development of biocrust restoration, is very high. But as we can imagine, over the next decade, as we have opportunities to potentially commercialize this could potentially decrease. So this is just thinking about, as I go forward with some of the results, some of the trade-offs of the different types of inoculum that we developed. So the next 2 graphs are going to look at, we'll be looking at the biocrust growth response. So I'm going to talk you through this, a lot of this graph first, because it's fairly complicated. So again, what we have here on the y-axis is chlorophyll A, again, which is an index of biocrust recovery and photosynthetic activity. And these 2 sites, we looked at, the next 2 slides, I'll be showing a more silty soil, and then a finer textured or a coarser textured soil on the next slide. So this is Hill or Utah Test and Training Range, and this is Jornada Experimental Range, where we worked at close to Fort Bliss. So looking at the x-axis here, these are our range of treatments. And I will start over here on the left. These are our natural recovery plots. So we've heard this before, where we just disturbed the soil and we watched how quickly the crust recovered. So all of the rest of these plots were disturbed, and then we applied different treatments. I'm going to move over to these far right 2 bars, is that we have 2 soil stabilization strategies that we implemented. One was the implementation of straw borders. So you can think of these as small silt fences of straw that would stick up approximately 10 centimeters above the soil surface. And we also had the application of polyacrylamide. So these synthetic materials that are that are commercially available that are used to prevent soil erosion. So in these last two bars, this bar here is the polyacrylamide, and this bar here is the straw. But these were not inoculated. So what I want you to do is just like pulling out the big picture here is that we monitored this over 3 years, 2015, 2016, and 2017 at these different sites. And if you pull back and kind of squint at this, what you can kind of see is that there is no horse that is winning this race relative to the natural recovery. There is some indication here, and I'm going to point out in the green, is that, and this is maybe not surprising, is that the field collected might be doing a little bit better than this is the greenhouse grown, and these are the mixed isolates. So that's kind of mixed news, is that we have a limited response to inoculation and soil stabilization. Again, here on the Hornata site, what we see is a field collected and this stabilized straw soil here. So this is the straw borders. By 2017, in the third year, maybe we are seeing some promise. So what I'd like to point out here, this target is how much chlorophyll is in an intact biological, the range of chlorophyll in an intact biological soil crust. So we kind of use that as a target for restoration. And so as you can see here, our target is 15 to 20 micrograms per gram soil. And, you know, we're not even halfway there after 3 years. But again, this site is very different because it has lower chlorophyll A, and the range is highly variable and in the range of 18 to 25. So then moving on to our sandier soils, what's interesting again here, this is the same sort of setup where we have natural recovery here. And again, a little Kentucky Derby reference is that there's not a clear horse winning any race here. But what I do want to point out is that again here, maybe this field collected straw, again, it might be doing a little bit better than other treatments. And what we'd like to do is continue to monitor these over the longer term and see if we get a different answer to this story. I want to point out here is that this Hornata sandy site is really interesting because it was highly degraded and there just really wasn't much biocrust there. So that there was no field collected for inoculation. And notice this y-axis, this is very low. It's a very low level of chlorophyll in a very different biocrust community. And these were highly mobile sands. So what I want to look at now is asking the question and thinking back to those previous slides. I guess the mixed news is that we saw limited responses, but I think the good news was that we were able to use some of these synthetic stabilizing products that are available commercially to stabilize soils, and we didn't see any impact of those products on the biological soil crust recovery. So what I'm showing here in the next two slides, it's a little bit different of a setup, is that this on the y-axis, it's soil aggregate stability. So how stable is that soil surface? And the top value that we can get from this metric is a 6. And so what we see is that all of these intact biological soil crusts in both this hill site and the Hornata site are very stable and they have the highest value. So here we have the intact plot. The next bar over is natural recovery. And so you can see that in year 1, soil aggregate stability decline was very low at both sites after our disturbance and then started recovering in year 2 and 3. What I'd really like to point out is this, with these red dots, these are all the polyacrylamide application plots, each one of these. So what you can see is that in 2015, we applied these soil stabilizing products, and they are right around a 6 for both of the sites. So what's interesting and what happens over time is that in 2016 and 17, those products are supposed to degrade, which they did. And some areas just degraded and stayed at the same level the next year. Some treatments, this is the greenhouse grown, degraded and then recovered. And so this has been an interesting result for us, because we can think about how we can apply these soil stabilizing products, know that they will degrade in the first year, but then we may have the biological soil crust, we're giving them time to stabilize and potentially act as a biological stabilizer. So coming off of this experiment, what we wanted to do is that since we had these mixed results with these different inoculum types, we wanted to ask the question, if we gave biological soil crust everything they need, shade and water, can we do a rapid restoration experiment? So this is just showing that we did the small plot inoculation experiment. This was actually at a different site on the Colorado Plateau near Moab, Utah. And we just did the inoculation of these small plots at a highly, highly degraded rangeland site that had been degraded by livestock grazing over the past 100 years. We then watered and shaded these plots and asked the question, can we induce biological soil crust very quickly over a 4-month period? So I'll be showing you the results of that experiment. So the answer to that is that our inoculation with the shading and the water, that if we wanted to do kind of a labor-intensive biocrust restoration, is that this does show some promise. So I'll start here in this middle graph. This is a percentage of biological soil crust cover. We have no inoculation, a low level of inoculation, which was about 10% cover of the soil surface, and a high level of inoculation. These bars on the left here are without a soil stabilizer, and then this bar on the right is with a plant-based soil stabilizer. And so what we can see here is we did a really nice job in this case increasing the biological soil crust cover when we did give the biological soil crust the resources they needed. With this inoculation, we also inoculated with lichens. And what we did see here is that with that water and shading, the lichens were also doing well. And again, the good news here is that there wasn't really evidence that those soil stabilizers, which are important when we're thinking about restoring soils, is that they didn't inhibit biological soil crust growth. So just another same axes and an experiment here, just another few metrics of recovery. So we're going back to chlorophyll A. EPS is exopolysaccharides, and also aggregate stability, which is what you saw in the previous slides. And what we see here also, we get a nice increase over that very short 4-month period with inoculation and response in chlorophyll A. And interestingly, there wasn't a strong difference between, necessarily a very strong difference between the low and high inoculation. So we know that we can possibly just inoculate at lower levels and things might be fine. Exopolysaccharides are just those, that glue that keeps those soil grains together, that can also, the specific compounds that really can stabilize those soils. And what we see here also is that there is some indication, it's a little mixed, of the exopolysaccharide response. And what we see, and this is an interesting path that we're on now, is that these plant-based soil stabilizers can add polysaccharides to the soil. And finally here, what we see also is some increase here, especially with the soil stabilization in aggregate stability. So some conclusions from this work is that we do have some fast and effective methods to grow biocrust inoculum now from some limited field collected samples. And I think one of the more exciting things that is has been happening really over the last year is talking about how we can salvage soil crust from sites that are being developed, for example, for housing developments or any other infrastructure, that we can go in and salvage those biocrust communities and then re-inoculate soils for restoration. We do have these slower methods of lab cultures that from these mixed isolates in the lab, and I think this has commercial opportunity, but that will take some time to really further develop that technology. And what we did see is that biocrust recovery can be enhanced with the shading and inoculation. And you know, if you really need to think about doing a restoration project very quickly, and you have the time and the resources, there is a potential to do rapid biological soil crust restoration with a lot of intensive shading and irrigation, and the use of synthetic products. So some of the benefits to DoD, these are fairly novel approaches to biocrust restoration, and we're really thinking about how do we restore degraded drylands from the variety of human activities. And it's really important for the function of these dryland systems. So there's potential rehabilitation benefits to ecosystems that are impacted by these land-based training activities. And our future challenge, and it'll be interesting to talk about, is scaling these approaches to the landscape scale. And what I would like to close with is that this area of research has really taken off, and over the next 6 months, a special issue of Restoration and Ecology will be coming out with over 30 papers on biological soil crust restoration. So I would say to everyone also, stay tuned with some of this research. And if you have any questions, please feel free to visit this site, and also contact me at this contact information.
Great. Thank you, Nichole. And as a reminder to everyone, you may submit the questions by using the Q&A box on your screen. We have received many questions for Nichole, and we're going to go ahead and get started, but please feel free to keep those questions coming. Nichole, what types of soils would you target for biocrust restoration?
That's a really great question. So what we saw with some of our research is that the potential for recovery is different on coarse versus finer textured soils, and it just recovery is more inhibited on coarser textured soils than finer textured soils. So I would say that if you have a degraded fine textured soil that's a clay or silty soil, then I think, and you have enough propagules of crust nearby, you might want to just think about protecting that area and seeing what the natural recovery might be. And there's a lot of great, fast, rapid metrics to evaluate the biological soil crust community in the field. And if you aren't getting enough recovery, what you could think about is then going in with inoculation later.
Great, thank you. This is a question from the state of Utah. Can biocrust be salvaged and stored for reapplication? And if so, for how long can you store them? And what do you store them in?
Oh, that's such a great question. And we've been asking questions. We've been doing research on that. And again, in the special issue, that is coming out, there is a paper on this. And so what we're finding is that, so let me start with the first question of, can these be salvaged? And the answer to that is absolutely yes. And what this would require is going into a site that is scheduled for development, for example, and the best way to do that would be to kind of scrape that top layer of the biological soil crust community. And what we do is we just scrape that with trowels, and store them in 5-gallon paint buckets. And so what you would want to do is that if they are scraped dry, you just want to keep them open to the air so they don't grow any fungal organisms that you don't want. So I would store them with that they have enough air to air dry and release any moisture that's in the soil, and store them in a cool, dark place. So the question about how long how long are these viable? So what we know from museums is some of these biological soil crust organisms that were collected 100 years ago, you can add water and they'll start photosynthesizing. Although what we know is that if they can be used within 6 months, that's probably your best strategy. But what likely is to happen is that you would just have reduced function the longer that they are stored.
Thank you, Nichole. This is a question from UC Davis. Why did you choose chlorophyll A as a metric for measuring recovery instead of something like percent cover?
That's also a good question. So we actually did both, because the percent cover, as you saw in that second slide, percent cover of the biocrust community in tandem with chlorophyll A. So we really looked at both, and sometimes they are correlated, sometimes they aren't, but they are both important metrics, and we did measure both, but I was unable to present all of those data today.
Thank you very much. Question from the state of Utah. How much irrigation is required for reestablishment?
So what we did in that rapid restoration project is that we were watering, and this is a bit difficult for me to answer because that rapid restoration project that I showed was actually in the exceptional drought of last year. So in that case, we were watering quite a bit. We inoculated in February. So what we would recommend is inoculating soils in the fall, going into the winter, so they really can have that water. And I would say this is really for areas that experience wetter winters. And so you want to really think about looking at the weather and inoculate when you know you have moisture coming. And so we inoculated in February, and we were watering probably early on every 3 to 4 days with a watering hose, which probably was equivalent to about a 10-millimeter rain event in that range. So early on, I think you would want to water quite a bit, and then it also depends on what your weather is.
Thank you very much. A question from ESSRE. For the greenhouse growth, what was the optimum nitrogen, phosphorus, and potassium formulation used?
Oh, that's a really good question. And that detail, I do not have it in the top of my head, is in one of our publications that was recently published. And if we can, I would be happy to pass along that publication of that optimal amount of nutrients. What we did see is that nutrients mattered, but really not as much as the water and shading.
Thank you. Say you were cultivating algae for DoD biofuels. Would the lipid extracted biomass be a good candidate for biocrust inoculum?
Oh, that's a question I've never had before. So thinking about biofuel, so we've thought a lot about using biofuel facilities to start scaling up. cultivation of these isolated cultures, but I really haven't thought about the lipid extraction specifically. So if someone would like to contact me to talk more about that, I would be really interested.
Great, thank you. Another question from the state of Utah. Did your research show that exopolysaccharide is a better soil protectant than polyacrylamide? And if so, is this product available commercially?
So the two types of soil stabilizers that we use, so the first one in the first set of studies was it's compositionally, the polyacrylamide is really similar to Elmer's glue. And then in that second experiment, what we transitioned to was this product, the plant-based product, which is a psyllium husk. And I think that it wasn't the EPS, but the psyllium husk. What we're really favoring that now because it is a plant-based product, and it does show that it enhances the exopolysaccharides in the soil. And what we know is that having enough exopolysaccharides in the soil can really influence the water holding capacity, the soil, and the soil stability, obviously the soil stability. And so we're really moving toward this plant-based product now, and it's also fairly inexpensive to purchase relative to some of the other synthetic products that are used by Department of Transportation, for example, for soil erosion along road cuts.
Thank you, Nichole. And at this point, I'd love to pull Jim back into the discussion and ask you both, perhaps starting with Jim, about your advice on how you can scale up your smaller scale plot level restoration strategies to larger landscape efforts. Jim, would you like to start?
Yeah, it's a really good question and a difficult one. We have tried tilling in the mycorrhizal inoculum and then broadcast seeding, and that has worked. It's very expensive in terms, it uses a lot more of the inoculum, and putting out nurse plants where we use a small amount of inoculum and we get the most out of it, and by tossing it out on the turf and mixing it in, we're more likely to have much of it solarized and die. The inoculum, so it does work, and it's a question of the production. Can we produce enough inoculum to make that feasible? Another approach is to put it into rows, and if you're seeding if you're drill seeding, you can put it in with the seed mix, and I think that would be get the inoculum closer to where the viable seeds are. It might be a better intermediate plan. But this is a general problem which we're going to have to work on with native inocula, and the problem with native inocula is that when you grow it up, and it has a bulky carrier because you have to grow it up in a soil media. And so it's something that we'll have to work on in the future.
Thank you. Nichole, what is your opinion?
So I think with our different approaches I described, that scaling up the biomass for the cultured lab cyanobacteria, one thing that we've been thinking about and exploring is the use of biofuel facilities to be able to do that. And from that approach, the greenhouse grown, what we're really, and this is being done now by different groups, and my group is also starting this, is to, you probably saw those small containers that we were growing in the greenhouse. And what we saw with the greenhouse, we had some mixed results because we started getting some of these weedy microbes from the greenhouse. So what we're doing is now moving this to outside nursery facilities, where we're actually thinking about renting facilities, and one group is actually doing this, renting facilities from a local nursery and growing and irrigating biocrust outside at very much larger levels. But the challenge will then be how do we add this in the field? And I'm pretty excited about some of the precision restoration that that is really starting up now. So there is drone-based precision restoration where seeds are being 56-pound payloads are being added to sites in specific areas where seeding can be successful for restoration from drones. And so I've been thinking about, if we want to just kind of go really innovative here, can we think about adding mycorrhizae or biocrust to specific locations where we know they'll be successful from kind of aerial-based using aerial-based strategies.
Great. Another broader question for the two of you, maybe we'll start with you, Nichole. What is the cost associated with the inoculum approach that you've used?
So as you can imagine, the cost to the project was very high in inoculum development, and like any economic approach we are trying to drive down those costs by taking different approaches over the next 10 years. So right now, the development of inoculum in the lab is very expensive. I would not suggest to anyone to really take that route, but growing inoculum outdoors at a nursery facility is probably the least expensive way to go. And that is just, if you have access to inexpensive labor or undergraduates or very passionate young folk, that it's probably primarily in the labor and application costs.
And Jim, yeah, please go ahead.
Yeah, so I think I don't know the exact cost. I agree with Nichole that what we've been doing in isolating these fungi and then maintaining them in the lab is very expensive. And in our case, once they're isolated, they can be grown up in larger quantities, but it has to be done in sterile media and has to be monitored to make sure that they maintain their purity, and that's all difficult. You have to have knowledge of the system. So I don't know the actual cost. I know the one company that is producing our inoculum for distribution, Microbial, I don't even know how much they charge for it. So in the other question would be the amount of inoculum put on the site, which would get into that. So the economic feasibility of the approach I think it will change as we develop more of the inocula, but right now it may be inhibiting on some systems.
So it's to be determined.
To be determined, yes.
Well, to be determined. Thank you both for your presentations and for taking the time to answer questions. I would like to remind everyone that our next webinar is on Thursday, May 23, and it will feature two speakers from Aptum Federal Services on the Institute treatment of emerging contaminants to improve the effectiveness and reduce costs for DoD land management efforts. We'll start with Paul Hatzinger, who will talk about a novel reactive gas process for the remediation of Vadose zone source areas containing 123 TCP. And then we're going to conclude with Dr. Paul Koster Van Groos, who will discuss the evaluation of natural attenuation and institute treatment for 1,2-dibromo ethane treatment at a complex field site. So registration is open for this webinar and other webinars through the end of 2019. So please make sure to visit the SERDP and ESCCP webinar webpage to register for these webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it if you can please take a moment to complete the very brief survey that will pop up on your screen at this time. This concludes today's webcast. Thank you so much for your time.